Well, welcome back for the second part of our mutations lecture. In this video, we're going to talk briefly about different types of polymerases and how mutation rates differ between RNA and DNA polymerases. We're then going to talk a little bit about horizontal gene transfer, which is a mechanism used by bacteria and other microorganisms to pass genes among organisms within the same generation. Before we get into mutation rates with polymerases, I want to give you a brief overview on the more technical name for different types of polymerases. Oftentimes, polymerases are named not only based on their template, but also based on what they're going to make. And they follow this pattern shown here. So for example, this first part tells us what is the template that this polymerase is going to read. The second part of the naming tells us what the polymerase is going to make. So for example, we oftentimes refer to the human DNA polymerase as that molecule that makes new DNA. Well, the formal name for this is that it's a DNA-dependent DNA polymerase, or sometimes abbreviated a ddDP. We know that the human DNA polymerase makes a copy of DNA from a DNA template. Similarly, we might call the human RNA polymerase a ddRP, or a DNA-dependent RNA polymerase. We name it as such because it reads DNA and transcribes that DNA template into an RNA sequence. So why take the time to talk about those different types of polymerases? Well, it's because in the viral world, there are more than just DNA-dependent polymerases. In fact, many viruses contain RNA genomes, and so they're going to require RNA-dependent polymerases in order to replicate their genomes. And what we find is that the mutation rate, or the spontaneous mutation rate by different polymerases, can vary quite dramatically. So what we're looking at here is a figure from a review that was published back in 2010. On the y-axis, we're looking at mutation rate. The higher mutation rate is towards the top of the y-axis. Lower mutation rate is on the bottom of that y-axis. And on the x-axis, we're looking at the size of genomes from small to large. Over on the lower left-hand corner here in our key, you can see different types of genomes marked with different types of symbols. But what we're going to do is we're going to take a look at these a little bit differently. Instead, we're going to look at these by the polymerase that they encode. So for example, these three here that have single-stranded RNA genomes or double-stranded RNA genomes contain an RNA-dependent RNA polymerase. This is an RNA polymerase that uses a copy of RNA as a template. Retroviruses, like HIV, contain reverse transcriptase. This is also the specific name for what we call an RNA-dependent DNA polymerase. In other words, it takes an RNA template and turns it back into DNA. It reverse transcribes it. And then at the bottom here in blue and black, we're looking at viruses and bacteria that encode a DNA-dependent DNA polymerase in order to replicate their genome. Now, don't forget that these organisms are also going to contain DNA-dependent RNA polymerases that are required for transcription. So when we map this by the type of polymerase, we find the following pattern. Those with RNA-dependent polymerases have quite a high mutation rate. Those with DNA-dependent polymerases have lower mutation rates. The other thing that you can see here as well is that as the mutation rate decreases, it allows for larger sized genomes. And so there's a lot of information in this figure. First, it tells us that RNA-dependent polymerases, particularly RNA-dependent RNA polymerases and reverse transcriptase, have really high mutation rates. The mutation rates of these polymerases are somewhere on the order of 1 mutation for every 10,000 nucleotides. For DNA polymerases, the mutation rate is lower, somewhere on the order of 1 in 10 to the 6th for viruses to 1 in 10 to the 10th or 1 to 10 to the 9th for bacteria. This is incredibly important because what this tells us, remember, mutations are one of the main mechanisms by which organisms evolve. Well, viruses that have RNA genomes undergo evolution and undergo selection at quicker rates than what we find within viruses that have DNA genomes. And for this reason, this is one of the major reasons why we right now, as of 2020, have to have a new influenza vaccine every single year. In contrast, if you've by any chance received the vaccine for human papillomavirus, you will likely only need 2 shots in your life for that, and it will provide you lifelong immunity for the rest of your life. And again, that's because the mutation rate in these organisms is so different. RNA viruses, because they mutate at a faster rate, are much more like a moving target when we try to treat them or to prevent infection. Whereas viruses with DNA genomes are much more stable. The other really important point, again, is that the mutation rate limits the genome size. Organisms with faster or greater mutation rates are going to have smaller genomes. The smaller size of these genomes increases the likelihood that functional viral particles will be produced without containing mutations in them. To finish off this section, I'd like to talk a little bit about horizontal gene transfer. Vertical gene transfer is the transmission of genetic information from one generation to the next, in other words, from mother cells to daughter cells. But horizontal gene transfer, on the other hand, is the transmission of genetic information from one cell to another within the same generation. There are 3 main types of horizontal gene transfer that we find in prokaryotes. The first is called transformation. In this particular example here, what we see is a piece of DNA from the environment coming into a cell. That DNA must contain homology, or in other words, areas of similarity to the genome of the bacterium that is shown there in blue. And finally, recombination has to occur to allow for the DNA from the environment to become part of the bacterial genome. The second form of horizontal gene transfer, which is in some ways similar, is known as transduction. In transduction, DNA from another bacterium will arrive to this new host bacterium by way of a bacteriophage, or a bacterial virus. Similarly to what we saw in transformation, both homology and recombination need to occur in order for this DNA to be incorporated into its new host. The third and final way is known as conjugation. Conjugation requires 2 cells that at the end of this process will both be living. It requires what we call an F positive and an F minus cell. The F positive cell carries what's known as an F plasmid or a fertility plasmid. And this fertility plasmid may encode on it genes for antibiotic resistance or for pathogenicity, but definitely it's going to encode for what we call a pilus, which is a hollow tube-like structure that allows for this F plasmid to pass through. And so what's going to happen here is that this F plasmid is going to be nicked on one of the 2 strands of the DNA. The strand that has been nicked will be passed from the F positive to the F minus cell. And after replication, both of these cells then will have an F plasmid. In other words, both of these cells then are capable of performing conjugation in future generations. So to summarize this point, why do we include this conversation of horizontal gene transfer when we talk about mutations? Well, it's kind of a wholesale mutation. It's not because of spontaneous mutations or mutations triggered by mutagens like we saw in our previous lecture. Instead, this is causing genome-wide changes, like we find in mutations, by bringing in genetic information from other cells. Unlike vertical gene transmission, which we often think to have subtle effects over time, these forms of horizontal gene transfer can have quite dramatic effects on an organism within a single generation.